So we will discussing about the Staphylococcus, and this Staphylococcus we discuss about that we were discussing about their pathogenesis. So I told you that there are three types of disease or infection this bacteria can cause. Either this Staphylococcus aureus enter into the body, it invade into the body as a whole, and then it cause a disease. Or second, this bacteria can produce toxin, and that toxin is ingested by the human, and there will be a gastroenteritis or GIT related symptoms and sign and symptoms. And third, bacteria can invade into the body, and after invasion the bacteria start producing the toxin. So three kinds of infections, there are three possibilities: invasion, like bacteria as a whole enter into the body and cause and colonize and cause infection. Second, bacteria is present in the food, contaminated food, but the bacteria has produced the toxins, so toxin is already present in the food. So when the patient eat the food, when the person eat the food, the toxin will cause toxicity and the patient will develop gastroenteritis. In this type of intoxication, this one, even when we try to find out the bacteria, like because bacteria is causing the food poisoning, so even if we take a stool sample, even if we take a stool sample to isolate the bacteria, so maybe the bacteria cannot be isolated because the gastroenteritis is not causing by the bacteria, but it is caused by the toxin. So we can isolate the toxin, but it is not always that you always get the bacteria as well in the stool. While the third type, we have both or mixed infection, infection plus intoxication. So bacteria enter into the body and it colonized, and after colonization it actually lead to a colonization, it produce toxin, and that toxin will cause toxic syndrome, or maybe it may cause scalded skin infections. So I think we have discussed about the epidemiology last time. So it can be transmitted from one person to another person. There is a direct transmission. Okay, so direct transmission, like if you are, because these bacteria are present on intimate objects or these fomites. So if somebody touches the surface of any these fomites, then the virus, this bacteria can be transferred. So this bacteria can also be transferred after eating the contaminated food. So let's start from the pathogenesis. So this bacteria, it has various virulence factors. It has various virulence factor, and as we have discussed earlier, that the virulence factors, they are actually the genetic, biochemical, or structural features of the organism. These are actually the structural features, biochemical, certain biochemical compounds that enable any bacteria to cause a disease. So these virulence factor, they are present within the bacteria, or either these virulence factors are produced, for example, present in the bacteria like capsule. Capsule is one of the virulence factor, or it may be produced and secreted by the bacteria as an exotoxin. So these are actually the virulence factor which cause a disease. So whenever bacteria invade into our body, the disease, you can say, the severity of disease, it depends on the type of virulence factor. If bacteria produce multiple virulence factor or a strongest virulence factor, the disease will be more severe. So the severity of a disease, it depends on the virulence factor of the bacteria, like how much virulence factor it is producing and what are actually the mechanism of action of those virulence factor. So Staphylococcus aureus, we are discussing Staphylococcus aureus. So S aureus bacteria, it express many virulence factors. It has many virulence factor, and because of these virulence factor, this bacteria is unable to hide from the polymorphonuclear cells, or especially we call the neutrophils. Simply we call the neutrophil cells. So it exude, or you can say it hide from our immune response because of its virulence factor. One of the virulence factor we discuss about the coagulase ability. You remember I have explained you about the coagulase. So coagulase is an enzyme that help in plasma clotting. Okay, this is not D, this is C, clotting. So this coagulase enzyme, it is only produced by Staphylococcus aureus species. It is not produced by any other species of Staphylococcus. So this is identification point of this bacteria as well. Staphylococcus aureus, it is coagulase positive bacteria. So when it enter into the body, it release this enzyme, and what it cause? It will cause a clot formation. For example, when clot is formed, inside bacteria is present. Inside the bacteria is present, but outside we have a clot. So because of this clot, for example, this is a neutrophil cell. So because of the clot, the neutrophil cannot reach to the staphylococcus bacteria. So that's how this coagulase it helps from hiding from our immune response. So that is why this bacteria, I told you, it is antibiotic resistant. This bacteria, it actually hide from our is or escape from our immune response by this coagulase activity. And this coagulase also help the bacteria for antibiotic resistance, because this clot, once the clot is formed, the antibiotic cannot penetrate inside this clot. So that also helps the bacteria for helps the bacteria against antibiotics. Okay, so these are the different virulence factors which can cause majority of diseases. And as I told you, the pathogenesis of any disease that bacteria is causing, it depends on these virulence factor. So first of all, we have the cell wall virulence factors. So these factors are present in our cell wall or the outer membrane of the bacteria. So first of all, we have capsule. So capsule, it is actually a polysaccharide. It is made up of the polysaccharide, and there are like 5 to 8 different polysaccharide capsules have been identified. So in this bacteria, there are 5 to 8 different types of polysaccharide or microcapsules have been isolated. So it can produce either of these type of capsule depending on the polysaccharide. So this capsule, it has a layer. It has, because capsule is a mucopolysaccharide. It's a polysaccharide. So that helps the bacteria to resistance against the phagocytosis. So this capsule prevent the bacteria from phagocytosis. So when you remove this capsule, the bacteria will be easily killed by our own immune response or even by a simple antibiotic. So this thing we also have discussed. When we isolate a sample from the patient, when we isolate a sample from a patient, this capsule is present around the bacteria, because when bacteria is present in the body, it has to hide from the or escape from the immune response by producing capsule. But when we culture the bacteria in the lab, when we culture the bacteria when we culture the bacteria in the lab, the bacteria lost the capsule. Why bacteria lost the capsule when we culture the bacteria in the lab? Can anybody answer? Yes, simple question. Yes, anyone?
Because of lack of nutrients.
No, 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 no. In culture media, we already have like lot of nutrients. That's why the bacteria grow on the culture media. Because in culture media there is no phagocytic activity. There is no neutrophil in culture media. So when there is no culture media, then the bacteria don't require to form a capsule. Capsule is formed to escape from the phagocytosis, to prevent the cell from phagocytosis. So that's why when we isolate the sample from the patient, the bacteria which we isolate, when we culture the bacteria, like from the patient sample, from the pus, from the blood, or from any source, you will see lot of capsulated bacteria. But when you culture, subculture, like take the sample from here to another, take one bacteria from here and culture it on another petri plate. So now this bacteria know that there is no immune response. So the bacteria which is grown, that bacteria will not have a capsule, because here in culture media there is no phagocytosis or phagocytic activity. So bacteria does not require this capsule. So capsule is only present in the in the sample which we take from the patient. The next virulence factor, we have protein A. So protein A is a major component of the Staphylococcus aureus wall. This is the main component of a Staphylococcus aureus cell wall, and its function is it bind to the Fc region of IgG antibodies. IgG antibody. So when it binds with IgG antibody, it will exert antiphagocytic activity. So this term we call it anti-opsonin. Anti-opsonin mean that antiphagocytic activity. So protein molecule, it binds with the IgG antibody and prevent the phagocytosis. And third, we have fibronectin binding protein, or we can also call it FNBP, fibronectin binding protein. So this protein, they are present on the surface. These are the surface protein, and they bind to the mucosal cells and tissue matrices, or simply we call that the fibronectin binding protein. It helps to attach the bacteria with the surface, with the mucous surface. So when bacteria enter into our body, uh, it it attach with the surfaces, mucosal surfaces, for example nasopharynx, maybe in GIT, with the help of this fibronectin binding protein. And the next factor that is also help in coagulation, or a clumping factor, that is we also call the clumping factor. This clumping factor, it is actually the same fibronectin binding protein. It helps to form a clump. Clump mean clotted particles, those are known as clumps. So these clumping factor, it helps the bacteria to form a clump. So when the bacteria is present in the form of clump, so there will be no phagocytic activity. So these clumps, these aggregation, it prevents the bacteria from phagocytosis, as well as these bacterial clump also cause antibiotic resistance. So this is clumping factor. The next we have the cytolytic exotoxins, the toxins which are produced or secreted by this bacteria, exotoxins. So there are four different types of like exotoxins: alpha, beta, gamma, and sigma toxins. So these toxins, they like their main, you can say, they attack on all type of cells, even RBCs, even RBCs. And the toxin which cause hemolysis, which cause hemolysis or breakdown of RBC, that exotoxin is known as hemolysin. So these alpha, beta, gamma, and sigma toxins, they actually attack on the membranes, cell membranes. And when they attack on the cell membrane will be lysed, and cell will be broken up. Like when the cell membrane is lysed, so it means all the cell organelles will be released out, so cell is totally destroyed. So among this alpha, beta, gamma, and sigma, alpha toxin is the best studied, like it has been studied very well. So it can polymerize, and it can pierce the membrane, break down the membrane. It can break down the membrane. For example, this is a normal cell. So this is alpha toxin. It can cause piercing, just like Caesar, we cut down something with the help of Caesar. So this alpha toxin, it pierce or it cut down the cell membrane. When cell membrane is cut it down, this is a nucleus and these are other cell nuclei. So what will happen? There will be a cell lysis. So that's how this alpha toxin is cause membrane lysis or cellular lysis. So this is one of the main pathogenesis of these cytolytic exotoxin. Then we have this pentone valentine leukocidin. Pentone valentine leukocidin. This toxin is actually a pore forming toxin. This toxin is a pore forming toxin. And this pore forming toxin, it form a pores on polymorphonuclear cells or neutrophils. What do you mean by this? Like this is a neutrophil cell. So it just like cytolytic exotoxin alpha, beta, gamma, this pentoline valentine leucidin, it actually make a small holes on a cell membrane. It make a small holes within the cell membrane. So by making these pores, ultimately it will cause the lysis, cell lysis. So this pentone valentine leukocidin and toxin, it is highly toxic, and it make this bacteria more virulent. And most bacteria which are MRSA, you remember yesterday I explained you about this MRSA. So this MRSA infection or methicillin resistant bacteria, they have this pentone valentine leukocidin factor or toxin. So these toxins are produced by these MRSA strain. So that is why MRSA is very, you can say, virulent as well as resistant against the immune response as well as the antibiotic. The next we have the super antigen. Here, I think this picture will help you to understand more easily. So super antigen, these antigens, they have an ability, they have an affinity for T receptor major histocompatibility complex class 2. You have studied this immunology, so you should know that this MHC protein which are present on the surface of T cells. So these toxins, it binds with the major histocompatibility class 2 receptor and form a MHC class 2 antigen complex. This toxin, it binds with the this toxin, it binds with the MHC class 2 antigen and form a complex. When this complex is formed, it increase the activity of T lymphocyte. It increase the activity of T lymphocyte. So actually it enhance the activity of T lymphocyte. So in normal immune response, in normal response, when antigen bind, in normal immune response, when antigen bind with the T cell, there is a production of T cell response. What is the T cell response? Release of inflammatory mediators like interleukin 2 alpha, interferon alpha, tumor necrosis factor. These are the cytokines which are released when T cells is activated. So when this toxin, which toxin? The toxin of Staphylococcus aureus, when it attach with the T cell, it increase the immune response up to 20%. In normal condition, when T cells is activated, it produce about 0.01 to 0.1% response. But in this condition, when this antigen, this toxin, it binds super antigen exotoxin, when it binds with this T cell, it enhance the activity up to 20%. So you can see here, there is a increase response, 20% increase in response. And so when there is a excessive response, what will happen? There will be a extensive release of inflammatory mediators. Excessive release of interleukin 2, interferon alpha, necrosis factor alpha, interferons gamma. So these cytokines, these are inflammatory mediator. So what will happen when these enzymes are released, these cytokines are released in high quantity, that will lead to produce a extensive immune response, and the patient will develop a toxic syndrome, toxic shock syndrome. So toxic shock syndrome, it is caused by this super antigen, and this super antigen, this shock is actually major factor for this is this super antigen. It increases the activity of T lymphocyte. It enhances its activation. Yes, class, understood? Any question? Yes. Okay, so till now we discuss about the different factors which are present on the cell. We have discussed about the exotoxins, like which this bacteria secretes. Now we will discuss about the enterotoxin. So enterotoxins, these toxins, they are released in the GIT tract. So that's why we call them enterotoxins. So enterotoxins, there are 6 major antigenic type. Due to their antigenic activity, there are antigenic variation. There are 6 major antigenic types: A, B, C, D, E, and G. And this Staphylococcus, it produce about half of them. Half of this enterotoxins, they are produced by this Staphylococcus aureus species. So how these enterotoxins are produced? So whenever the bacteria, it come in contact with the food, it grow in the food. When it grow in the food, it secrete this enterotoxin. And when a person eat the food, then this enterotoxin enter into the body and cause a food poisoning. It cause a food poisoning because this disease, it cause um, this gastroenteritis. because it is causing gastroenteritis. So in gastroenteritis, there is also a vomiting as well, vomiting and diarrhea. Vomiting and diarrhea, these are the main symptoms, plus fever. These are actually the main, you can say, constituents or the sign and symptoms of this food poisoning. So diarrhea is caused by locally, like in GIT, but the vomiting is caused by the stimulation of chemoreceptors trazodone CTZ, chemoreceptor trazodone in the CNS. So this toxin, this enterotoxin, it enter into the blood, reach to the brain, where it stimulate the chemoreceptor trazodone. This chemoreceptor trazazones is this part of the brain is responsible for vomiting, and this center is also known as vomiting center. So after ingestion, this toxin, it invade and it stimulate this vomiting center, and because of this vomiting center, because the activation, the vomiting will occur. So this enterotoxin, that's how it causes vomiting and diarrhea. So this toxin, it is a type of super antigen, and these enterotoxins, they are heat stable. Like even if you re-hot, like your food, for example, you bought the food or you cook the food and you put it for some period of time in some open area, like without covering, you just leave the food. uh, so if the bacteria is present there, so bacteria will grow in the food and it will secrete this enterotoxin. And when you re-warm the food or reheat the food even after using microwave oven, so the bacteria will be killed by microwave oven, because microwaves are the radiation, but the enterotoxin will still be there because it is heat stable. So when you eat that food, this toxin will enter into the body, and ultimately you may develop gastroenteritis gastroenteritis or diarrhea or vomiting. So that's how this enterotoxin, it it actually cause diarrhea, vomiting. Okay, any question?
No, sir.
Okay, so next we have the toxic shock syndrome toxin, or we can call it TSST1, toxic shock syndrome toxin 1. And we also have discussed about the antigen which are causing this toxic shock syndrome. So because this toxin, toxic shock syndrome toxin, it has the same similarity in molecular structure which cause this toxic shock syndrome. So this Staphylococcus enterotoxin F, it is actually the main toxic shock syndrome toxin. So the Staphylococcus aureus toxic shock syndrome antigen is Staphylococcus enterotoxin F. So this enterotoxin F, as the name suggests, enterotoxin. So it should have to cause diarrhea and vomiting. But instead of causing diarrhea and vomiting, this enterotoxin toxin F, it cause immune response, it cause shock syndrome. So it does not cause any kind of diarrhea. So it does not cause any food poisoning. So this toxic shock syndrome, it is caused by enterotoxin F. Okay, so this is actually the factor. It is a toxin for shock syndrome for Staphylococcus aureus bacteria. The next we have exfoliatin. So exfoliatin toxin, this toxin, it causes exfoliation. It is also a super antigen, and it is responsible for causing scalded skin syndrome. Scalded skin syndrome. And what is the mechanism of action of this toxin? This toxin, it actually break down the dysmoglin 1. Dysmoglin 1, it is actually the component of desmosome. It is, yeah, it is the component of desmosome. And what is desmosome? Desmosome, it is actually the adherence. Just like, we have adhered chemical we use to join two surfaces. Same desmosome, it act as a adhesive material which cause cell to cell adhesion, so that the cell, it remain intact with each other, it remain attached with each other. So when you remove this desmosome, so cell will not adhere, so there will be a cleavage, so skin layer will be separated. This is one layer of skin, second layer, third layer. So if no desmosome, the layers of the skin uh, will be separated. That is we call exfoliation. So this exfoliation, it is caused by this exfoliating toxin. Okay, class, we have less than a minute. After a break, we will continue.